students, in this video of ours, we are going to learn about the second application of DNA biotechnology, that is DNA recombinant technology in the field of medicine, and that is gene therapy. Now, this gene therapy is when it generally means that when we are replacing the faulty gene, which is creating any sort of disease in the body, we are replacing that faulty gene with a normal functional gene, and that is called gene therapy. So gene therapy is an attempt to deal with genetic or congenital diseases. What is congenital diseases? The disease which is present since birth. Since birth, the child is having that disease. It's a genetic the disease which is due to some faulty gene, or which is due to absence of gene. The gene is not present, it has been deleted, and so the relevant protein is not been formed, and so the disease has been there. This aims at correction of a genetic defect by delivery of a normal gene into an individual or embryo to take over or compensate the function for a non-functional gene. That means what is the aim of the gene therapy is we are replacing the faulty gene with the normal gene from outside, so that the relevant protein can be formed. Now, the first disease to have a gene therapy, the first clinical trial was done for gene therapy was done for the disease which is adenosine deaminase deficiency disease. Now, as the name suggests, in this disease there is deficiency of adenosine deaminase. Now, adenosine deaminase is the enzyme which is formed from a gene. So the gene which is responsible to produce that enzyme is being deleted or has become non-functional due to any kind of reason, and so the adenosine deaminase enzyme is not being produced in the body, and that disease is called adenosine deaminase deficiency disease. Now, in this gene, coding for enzyme adenosine deaminase gets deleted, leading to deficiency of ADA and problems in immune system. Basically, this adenosine deaminase enzyme, what is this enzyme doing in our body? In a normal person, this adenosine deaminase is helping in the formation of lymphocytes B and lymphocytes T from the bone marrow stem cells. So, from the bone marrow stem cells, the lymphocytes A and B are formed, and they are formed in the presence of this enzyme, adenosine deaminase. Now, suppose adenosine deaminase is not there in the body. So, in that case, in that person, the stem cells will not be, bone marrow stem cells will not be able to form the lymphocytes B and T. And we know that lymphocyte B and T are responsible for humoral immune response and cell-mediated immune response of our body. So in that case, both immune responses will not be there in our body. So that person or that child will not be having any immunity. So there will be problems in the immune system, because humoral immune response as well as cell-mediated immune response is not there because of the deficiency, or because of the deficiency or not being present of this enzyme of adenosine deaminase. And this is because of deletion of that particular gene which is responsible for production of adenosine deaminase. Now, this adenosine deaminase deficiency is treated by earlier, before the gene therapy was discovered or introduced, it used to be treated by bone marrow transplantation. So bone marrow used to be, they used to transplant the bone marrow into the patient's body so that the lymphocyte B and T can be produced in the body with the new bone marrow. Or the enzyme replacement therapy used to be there, where the enzyme was replaced, the enzyme was given into the body. Now this gene therapy for the ADA deficiency, this was the first clinical gene therapy which was given in 1990, and it was given to a 4-year-old girl who was having ADA deficiency. Now what they did was, they isolated the lymphocytes of that girl. Okay. Now these lymphocytes, they were isolated from the patient's blood and they were cultured in vitro. That means in blood, and they cultured those lymphocytes in vitro. In vitro means under laboratory conditions, these lymphocytes were cultured. Now this functional ADA, C is what? Complementary DNA. Now this functional ADA complementary DNA, that means the DNA which was responsible for the formation of ADA, adenosine deaminase enzyme, was introduced into the cultural lymphocytes. How it can be introduced? It can be introduced by using a vector. And here they used a retrovirus as a vector. So by using a retroviral vector, this gene of interest. Now what is our gene of interest? That is the gene which is responsible to produce adenosine deaminase enzyme. So this gene was introduced into the cultured lymphocytes which were there in vitro, which were cultured in the vitro. Now these lymphocytes, once that DNA, the gene was incorporated inside, these lymphocytes were again returned back to the patient's body. Now what happened? The patient body responded and the immunity improved. But what happened was that these lymphocytes are not immortal. The life of lymphocytes is not for the lifetime, because they keep regenerating in our body. So what will happen? In this case, you need to infuse those lymphocytes repeatedly into the patient's body. So lymphocytes are not immortal. Therefore, repeated infusion of genetically engineered lymphocytes are required. So what is the permanent cure? Permanent cure is that if we can introduce this gene from the bone marrow cells producing ADA into the cells at the early embryonic stages. Early embryonic stages means suppose when in the zygote, or when the embryo is 2-cell, 4-cell stage. Early embryonic stages, if we can introduce this bone marrow cells which are producing the ADA, which are having the genes which are producing the ADA, then it can be a permanent cure for the lifetime, because that bone marrow stem cells are going to produce the lymphocytes B and lymphocytes T in the patient's body. So that is the permanent cure. But now, because this was introduced in a 4-year-old girl, so the regular infusion was required. So when there are patients who have been treated after, because this was not, this therapy was not there earlier. It was introduced only in 1990. So there are many patients presently who are taking this gene therapy. So they are regularly taking it for this ADA deficiency because the lymphocytes are not immortal. So regularly they have to go, and then the lymphocytes from their body are taken out. They are cultured outside, and then they are with the help of the retrovirus, the desired gene is incorporated inside those lymphocytes, and then these lymphocytes are kept in are sent back to the patient's body, and then they can produce the immune response. But the permanent cure is also not now being discovered, that if we want a permanent cure, then in that case, this gene should be introduced at the early embryonic stages, when the stem cell are not yet been differentiated into lymphocyte B and lymphocyte T. See, this is how it happens. This is the patient's body. First of all, the first step is that cells are harvested from patient. Cells have been taken out. Lymphocyte cells have been taken out. Then now here what you have done is, in lab, the retrovirus is there, and retrovirus is bringing the desired gene, and this desired gene is then sent to the lymphocyte with the help of this vector, and now the desired gene is inside the lymphocyte. Now what happened? The altered virus mixed with the patient cell. Now this patient, this is the altered virus which is having the desired gene. The desired gene is inserted into the virus. Now this is the altered virus. Now when we put that virus inside this desired gene will get incorporated with the lymphocyte. And now this lymphocyte is having the desired gene. And now again these lymphocytes can be put back into the body of the patients. So now these lymphocytes are capable of producing, these lymphocytes are there, so they are capable of producing immunity response. So immune response is possible in these patients with the help of the gene therapy.